Okay, this is the first of two videos for this week. This is 6.1, outlining material from Chapter 9, Cell Division and Mitosis. In this video, I'm going to talk about normal cell division, but I'm going to save all of this cancer stuff for the next video when we cover Chapter 10, which is mutations and cancer. The cell cycle is how normal, healthy cells replicate. Most of the time, the cell is not actually dividing or replicating. It's in a phase called interphase, which is the majority of this pie chart of the cell's life cycle here. Sometimes you hear this called a resting phase, but that isn't accurate because a cell is always pretty busy even when it isn't replicating. All cells have some function, and in multicellular organisms like humans, individual cells have a very specific purpose. So think about like a muscle cell. Even if it's not dividing, it's active, it's doing quite a job. It needs to contract and stretch, and there's nothing resting about that, even if it is an interphase. Interphase has these three main parts. There's the G1 phase, the S phase, sorry, I covered that up, it says S, and G2 phase, and I will keep using those, so try to remember just G1 first, then S, then G2. The G stands for gap if you want to know, and I don't remember what the S stands for. Okay, so most of the time a cell is actually stopped right here, right in G1, or sometimes you'll see like G0, which is right before even the cell is doing much of anything. It's just halted there, and then it'll start doing some producing new organelles and things like that when it's getting ready to divide. But I'll just talk about G1 to simplify things. So in the S phase, this one is important right here. This is when DNA is replicated, all right? This diagram over here shows the chromosome perspective. It's really quite useful, this little piece, because this is showing how that DNA is divided. So we see that during G1 part of interphase, there's just this one chromosome in the cell. Each cell just has one copy of each chromosome, but remember that we have pairs of chromosomes. We're what is called diploid. Your book talks about that in one of these chapters in this unit. I think it's a little bit later on, chapter 12, I think. But anyways, we have pairs of chromosomes. A human being has 23 pairs for a total of 46 of these chromosomes. During the S phase, each chromosome in each pair makes a perfect replica of itself, and they remain attached into this X-shaped structure. Each side is a sister chromatid, and they're attached at something called the centromere. Those sister chromatids are identical to each other, barring any small mutations that may have happened, because those do happen periodically, but otherwise they are identical. So then during mitosis, which we're about to get to, they are actually torn apart and distributed into two identical daughter cells, so that each cell ends up with a complete genome and starts again here in the G1 phase with its entire genome in one copy. So cell division through mitosis, once we come around into here, it can happen for one of three reasons. It can happen because a body is growing and cells need to divide. It can happen because a body is injured and needs to heal a wound, or just normal cell replacement. So there are cells that wear out and you need to keep making. Your skin, you may have heard that you grow a new skin every 6 weeks. What that's talking about is your outside layer of your skin is there to protect you. It's there to kind of sacrifice itself as a barrier to scrapes and sun damage and things like that. Those cells get worn out, so they slough off and get replaced. And it's about every 6 weeks that you basically have new external skin. You still get wrinkles and things because it's the dermis underneath doesn't get replaced, and so it can, stretch out and get wrinkly and things like that. Red blood cells are also kind of weird, so they get brought up as an example to a lot of things. Remember, red blood cells don't have a nucleus, which means they can't make proteins, and they can't fix anything that goes wrong with them. They eventually start to deteriorate. Cells don't last forever. And so about every 120 days, your red blood cells get completely replaced as well. Okay, so mitosis itself has 4 individual phases within it. We're going to go through each one of those individually. The first phase in mitosis is called prophase. If you recall from the last couple of videos, I told you that DNA is not usually supercoiled into these chromosomes that look like this, sort of. Most of the time it's not like that at all. It's in this semi-unraveled chromatin structure. Remember, the individual strands are wrapped around proteins called histones, and they form some complexes, but it's not supercoiled into a chromosome because genes need to be accessible so that mRNA can be produced and genes can be expressed. This is also true for replication during the S phase. So during the S phase, that unraveled chromatin is replicated here in the S phase. Afterwards, after that happens, you now have 2 copies of that chromosome, and it will supercoil at this point. So you get something kind of like this, and I want to try to stick with my color coding. Bear with me. So now you have these sister chromatids, and they're attached here with something called the centromere. I think I mentioned that in the last slide. Okay, so now you've got replicated DNA, and you've got 2 identical sister chromatids for each chromosome. At the same time, during prophase, the nuclear envelope, which is surrounding this is all in the nucleus. The nuclear envelope is neat because it's actually 2 lipid bilayers. So each lipid bilayer is 2 layers of lipids. The nuclear envelope has 2 of those. And I didn't draw it in, I should have. It also has these nuclear pores where messenger RNA can enter or can exit the cell, and actually certain things do enter, but we'll leave that for another way. So this starts to deteriorate, and so you end up with something that looks like this. These pictures, by the way, show plant cells on the top and animal cells on the bottom. They have to be stained different ways to look at them under a microscope, so that's why they have different colors. Okay. So also, real quick, these microtubule fibers begin to form the mitotic spindle. That'll be very important in the next phase. Metaphase is when these replicated chromosomes line up, oh, beautiful, in the middle of the cell. It's really quite, quite easy to spot once you know what you're looking for. So there are more of these. They're just lined up in the middle of the cell like this. Here in this plant cell, you see the chromosomes are in blue. The mitotic spindle is in green. It's really easy to see in that stained plant cell. Anyways, so what happens here, let's make, what did they use, green for the mitotic spindle. The mitotic spindle is attached at either pole of the cell, and it grows out and it attaches to the centromere of each chromosome, and what it's going to do is it's going to tear these sister chromatids apart. So it attaches right here in the middle on each side, and it's going to just tear them apart. So this mitotic spindle attaching to the centromeres of each chromosome is what causes them to line up near the middle of the cell like that, in this nice orderly fashion in preparation for the next phase. So in that next phase, called anaphase, these sister chromatids are now ripped apart by these microtubules, this mitotic spindle, which does this just by shortening. So that mitotic spindle, just the microtubule just starts to shorten, and that just rips the chromosomes apart. So what you end up with here is on each side you have an entire copy of the genome. Each sister chromatid is now torn apart, and you have those nice individual chromosomes like you had before. So an entire copy. Once they're back on that one side that they need to be in, this is really fun, on either side, the nuclear envelope reforms in this phase called telophase. I've heard people say telophase too, doesn't matter. So now you've got, for just this moment, you've got a cell that is binucleated, or has 2 nuclei, which is kind of neat. Some texts will include and some will exclude cytokinesis, which is when the cell membrane out here begins to pinch in and it separates the cytosol and the organelles within that. So you're going to end up very quickly, these are going to pinch together like 2 balloons or something, you're going to end up with 2 daughter cells. That's cytokinesis. I like to talk about it with telophase because it makes more sense to me, and you have to remember too that nature actually doesn't care that we like to organize things into these discrete categories. It's all really one continuous process. We assign a beginning and end to each of these phases. It doesn't exist actually in nature. Now, if you're a plant cell, like up here on the top, remember these are plants, you have to do an extra step because I think it's this red in here. It's really hard to see with this staining, but this red is cell wall. You see it's not very clear in this dividing cell. But in a plant cell, the plasma membrane will pinch off like this just fine, but then there's this cell wall out here. Oh, that's a terrible color for this. Let's go with bright blue. This cell wall out here has to also replicate. So what it does is it just starts growing this way, and you end up pinching it off with that extra step of just growing that cell wall. So this is anything with cell walls. Plants, fungi have cell walls, bacteria have cell walls, but they actually don't go through mitosis. It's something else. It's the same idea, but it's called binary fission. Point is, they have to divide those cells with a new cell wall. Okay, hopefully that was short and sweet. There's a lot of information in there, but what I want you guys to be able to do is to apply it. So, after learning about mitosis, what the heck does that help us to understand about GMOs, which is the relevant topic that I'm assigning for this unit? So I want you to consider this. You have 2 daughter cells from a cell division that are identical. Again, there might be 1 or 2 mutations, but pretty much the exact copy of each other. Now, if one of those mutations does happen, and then that cell divides, each of its daughter cells is also going to have that mutation. But the other cells in whatever organism, they don't necessarily have that mutation unless they're all descended from a single cell that had that mutation and then divided. But one mutation in one body cell does not necessarily mean a mutation in the entire organism. So if you want to introduce a trait, and remember this is more than a point mutation, a trait is an entire gene or even a collection of genes, you want to put that into an organism, you want that entire organism to produce or to have this gene. So if you want a goat to produce antithrombin protein, you want to make sure every cell in that goat has that gene so that you can make absolutely certain that all of the mammary cells in that goat express that gene so that you can get as much antithrombin protein from that goat as you can. what would you do? Which cell would you change? I want you to also remember that individual organisms do not become transgenic from something in their environment after they've grown. Transgenic organisms are born that way. Lady Gaga sings a whole song about this. This is because new genes that are spliced into a cell, they have to be spliced into an egg cell, and then that egg cell divides, and you end up with an organism that has that trait in all of its genes, or all of its cells, I'm sorry. Remember, each cell has an entire copy of the genome, but only certain genes are expressed in certain cells. That's a topic that we'll talk about with stem cells in chapter 13, I believe, how cells differentiate and only express certain genes. But anyways, okay, so your goat, or you have goats, you want to make them produce antithrombin, you've got to genetically engineer the egg, not a baby goat or a full-grown goat, an egg, because you want that goat to have that gene in all of its cells so that it can be produced in the correct cells. If you have, say, a corn crop that you want to be resistant to Roundup so that you can spray a bunch of herbicide and kill all the weeds and get a more plentiful crop harvest, then you want to make sure that the entire corn plant is resistant and it's not going to die. Or you want all of the tomatoes on your plant to be freeze resistant, so you engineer the seed that contains the egg. You get the picture, hopefully. Now, there is actually one other way to get a transgenic organism. You can genetically engineer a sperm cell instead of an egg cell. So I want you to just hold that into your mind for the discussion for this week. Alright, from this video lecture, I hope you understand a few things. There are three pretty much normal purposes for cell division or mitosis right here. I also want you to remember that there are these other phases in the cell cycle. So interphase is these three, and then mitosis as well. Mitosis itself has these other four phases within it. The result of mitosis is these two daughter cells that are identical to each other. After this video, you should go on and read chapter 10 if you haven't yet. Watch lecture 6.2. Alright, see you then.